Hello, I'm Dr. Vivian Lowe. Welcome to VLMD Rounds, a podcast on medical science and tools to optimize your health. It was a really packed week last week, and I think next week looks even more packed if that's possible. I'm just really glad I found some time to make this recording. Before I start, I just want to acknowledge a few people. So thank you, Anthony, for your comments on YouTube on the brain. So that was a nice little conversation that we had on shrunken brain syndrome. And then Vivek, you had requested an episode on insulin resistance and mental health, I believe. So I've put that on the list for sure. And I hope to be able to get to it, maybe in a few months. I love it when you write in, all of you, and send me your requests because it helps me kind of focus in on the topics that you're interested in. The difficult part is just finding the time to kind of line up all these different episodes, make the appropriate notes, and then actually film it. All right. But it's been almost 6 months since I started doing this, and I've made an episode for every week. So that's not too bad. Somehow we got that in. I hope I can keep up the production rate, but we'll see. Okay. Last time we started talking about the immune system, and the reason I've been going into the immune system is that people have asked me to talk about inflammation. We mention inflammatory and anti-inflammatory processes in the body a lot, and then there are lots of diseases that have inflammation at the root of those diseases. And then we also have autoimmune diseases. So there's a whole range of topics involving the immune system. I thought it'd be worthwhile just getting some basics in, the foundations in on the immune system. I just want to point out also that I've mostly been focusing on the cells of the immune system, the cellular part, because the immune system is vast, as I hope you're beginning to realize, and quite complicated. And there are all these special proteins and signaling molecules and different parts of the immune system that are involved that we're still learning about and making discoveries on. So by no means am I covering the entire immune system. I'm just going to be focusing, at least for now, on the cellular aspect. So last week we looked at mainly the cells of the innate immune system. We did talk a little bit about complement proteins and so forth, but ultimately we were focused on the phagocytes and killer cells of the innate immune system. This week we're going to look at the adaptive immune system. Let's go. For this week, let's just start off by kind of reviewing. The innate immune system, many of you already know, is your ready-to-go, ready-made system, so that if you're getting an infection right now, then there are ways that your body, those cells in your body, can recognize danger or some invader in your body through these standard molecular patterns and then activate so that they will start to defend the body against those pathogens. Now, this means that they can be deployed very quickly, but they're not very specific, and some of the cells of the innate system are sort of indiscriminate in their destruction, and so we get a lot of widespread tissue destruction if that's all we're relying on. Fortunately, we have the adaptive system, and with the adaptive system, this is more of your tailor-made system where we are now specifically targeting the pathogen or organism that is invading your body. So we're not going after general danger signals or pathogen signals. We're going after that specific organism. So if it's pneumococcus, if it's Pseudomonas, whatever the bug is, we are going to be specific for that organism. Now, I am only going to look at B cells today, because again, when I looked at my time, I just realized it would take very long to complete the whole adaptive side, the cells of the adaptive system. So today we are primarily just going to look at B cells as part of the adaptive immune system. Okay, so vertebrates have the adaptive immune system. Invertebrates don't, and they've done very well without it, but vertebrates are a little bit more sophisticated, so we've added this component to our immune system. So we have the adaptive system. Now, when we talk about the adaptive system, I think what comes to mind for most people would be antibodies. You know, with the recent pandemic, people think about, oh yes, okay, developing antibodies against certain organisms, against certain bugs. So these antibodies are special proteins that recognize specific substances. All right, just very generally, that's how we'll describe them. If you were to draw a representation of an antibody, I thought about how I would describe this, because if you're listening in, I want you to be able to visualize it in your head. So it would be kind of like drawing a martini glass. So if you had a piece of paper in front of you, you know, your antibody is going to look like a martini glass. All right. So you're going to draw, so you'll have one side that looks like half a Y on the left side of the martini glass. That's made up of the part that holds the liquid and then the stem as well, right? So it's kind of one half of a Y. And then you have another side which looks like the corresponding side of a Y, right? And so you have this martini glass shaped things. Those two sides of the Y would be what we call the heavy chains, okay, the longer heavy chains of the antibody. And then next to the sides of the glass, on each side, we would draw, you know, a parallel line to the sides of the martini glass, and we would call those the light chains. So that's how you would represent it if you were to just kind of sketch it out on a piece of paper. So you have those different regions. You have the light chains on the outside, the shorter ones, and then the whole sort of martini glass with the stem, and that part is the heavy chains. Okay, so those are the components of the antibody. And the part of the Y that consists of, you know, the shorter chain and then the top part of the heavy chain. So this would be the sides of the martini glass. That top part is the antigen binding region. And it's often designated as FAB, capital F, small ab. That's the region that binds to antigen. And then the stem part of the martini glass is referred to as FC, or the constant region. Okay, so that's the part that binds to FC receptors, and we'll talk about that a little bit later. All right, so you have two parts. First you have two different protein chains. You have the heavy chains and the light chains on each side. And then you have the top part, which would be the antigen binding region. And then you have the constant region below, which is the stem part of the martini glass. And we have different classes of antibodies that are appropriate in different contexts. Now, the whole point of the adaptive system is to be specific, right? So that we can recognize and attack a specific pathogen or organism. Now, first off, the antigen is really what is recognized by receptors, okay, of the cells. That's the antigen. The specific part of the antigen, the protein sequence of the antigen, which actually binds to those receptors, that would be called the epitope. E-P-I-T-O-P-E. So sometimes you hear about epitopes and antigens, and sometimes people use them interchangeably, but really the antigen is just whatever is recognized. Epitope would be the specific region that is recognized and bound to the receptor. All right, and it probably is a sequence of amino acids, depending on the organism. Okay. So how, because we have so many possible antigens out there in the world, so how is it that your body can respond and make an antibody that is specific to that antigen? So we have chromosomes, obviously genes, that encode for the heavy and light chains in those antibodies, okay? And you have one chromosome from your father and one from your mother. So you have 2 chromosomes, okay? And we have these sections, gene segments in them, or sections in them, that can be shuffled, okay, to make the heavy chains and also the light chains. So these segments are V, D, J, and C segments. Okay, so on these chromosomes, you have these segments laid out. Now you have different, possibilities of let's say the V parts. So you have 40 different types of V's, okay, that you could use. You have about 25 choices in the D's, and you have about 6 in the J's, and maybe 10 of the C segments, okay? So you can randomly pick and mix and match the different segments, right? So let's take a cell, a B cell, and it will pick one of the D's, one of the 25 D's. So let's say it picks D5, and that joins to J2, let's say. So now we have D5, J2, and that complex can then also, join to maybe V6, okay? So now we have V6, D5, and J2. Now another cell may have D1, J6 joined together, and then they're combining with V25. All right, so you can start to see there's a whole range of possibilities there for mix and match, and then they also can combine now with one of the C parts in the gene segments, and you have this sequence that will now be used to make that particular heavy chain. And it will differ between different B cells because, as I said, the possibilities, are quite wide open. You could use any of those combinations, right? So that's how you mix and match to generate diversity. And these segments, of course, have to be properly joined, the D to the J and the V and then to the C, right, to produce a full length and viable heavy chain protein, okay? If you don't join them properly, then it's not viable, and then that B cell just dies. We get rid of it. But that's okay because you just have so many B cells. So next, right, it's kind of like auditioning for a play or auditioning for a dance performance. So this combination doesn't work, next one, right? This dancer doesn't work, we'll use the next one. So, all right. So you have to be able to make a viable, functional, heavy chain, have those segments joined properly. If not, the cell just dies. We can get rid of it, right? But when you do get a functional heavy chain that you could use, then we make that heavy chain, and that's brought to the surface of the cell. And once you do that, you basically kind of commit the cell towards that particular antibody, okay, made from those particular gene segments that were selected out. So the light chains are made in a similar manner, and then they also make their way up to the cell surface. Now they have to also bind properly and align properly with the heavy chains to make a fully functional antibody at the surface there. If not, if they don't align properly, guess what? That cell just dies off, okay? So it kills itself, right? Again, next, right? So a lot of things have to align to make sure now we have this functional antibody at the cell surface. Now, I want to point out that this antibody at the cell surface, when it's attached and anchored to the cell surface, we call it the B cell receptor. But it's essentially the antibody, that martini glass structure. It's essentially that, but when it's attached to the membrane at the cell surface, we refer to it as the B cell receptor. Okay? When it's released into the blood free, it's not tethered or anchored to the cell anymore, then we refer to it as the antibody. All right, so that's the difference. One is anchored to the cell, and that would be the B cell receptor, and when it's free and floating in the bloodstream on its own, then you would call that the antibody, right? So each B cell has only one type of B cell receptor. It can make multiple copies of that, but it's that specific B cell receptor or antibody, right? Made from those particular gene segments that were selected out. Okay, now you have, lots and lots of B cells. And so you have a whole range of different antibody/B cell receptors that could be made, because those combinations, as I mentioned, are pretty endless, right? You could mix and match and get very different combinations. And so you have the possibility of recognizing potentially almost anything out there that gets into your body, okay? So we have that possibility, but each B cell that makes these B cell receptors, they're just going to make a few and pop them up on the cell surface and not really do anything except wait, okay? So what is it waiting for? It's essentially waiting around to see if we can put that B cell receptor/antibody to use, right? And if nothing happens, well, we don't need it, and over time that cell dies, okay? But if it's out there and it happens to meet its cognate antigen, right? So the cognate antigen is the antigen that matches that B cell receptor, okay? If that happens, then we can actually now utilize that specific antibody, okay? So you can see we have this potential and we use that, so we have a range of diversity in the types of antigens we could recognize, right? But if they're not actually there in your body, then you don't actually utilize them at all. But when you get infected by a specific organism or pathogen, for example, and that organism is the antigen to a specific B cell receptor, then we're going to rev up production of that particular B cell receptor, okay? So what happens then is we're going to prompt the cell to go into cell proliferation and cell division. So that B cell is eventually going to split into two cells, and those two cells will split up, and now we have something called clonal expansion, and we're actually making replicas of that specific type of B cell that's producing that B cell receptor. So at first we just have one of them sitting in there with a few of these receptors at the surface, But when it recognizes its cognate antigen, it's going to rev up production, make a ton of other B cells with that specific B cell receptor, right? So essentially now we get to the point where, okay, when we need it, we can make a lot of those B cell receptors, and eventually those receptors can get released into the blood. And at that point, those antibodies are used to recognize the antigens in the blood. All right. The antibodies themselves, just so you know, they don't actually kill the pathogen. They mostly opsonize or tag the pathogen. So if you remember from the previous episode when we talked about the complement system, we talked about opsonization, which is to tag organisms so that the phagocytic cells know these are the ones to kill, these are the ones to go after. Well, your antibodies can do that, and they bind to bacteria, to viruses, and so forth, tag them. They bind via the antigen binding site, the FAB sites, right? And then the FC portions, the constant regions, now bind to FC receptors in phagocytic cells like macrophages, for example, right? And then those macrophages can now destroy that pathogen, okay? So, and once the let's say the macrophage binds to that specific antibody, it sort of revs up its own phagocytic ability as well. So it's going to be a more voracious phagocytic cell. Okay? So that's one way that the antibody works. The other way is, viruses, they're kind of sneaky. For example, they like to get into cells, and they're going to utilize the cell's machinery to reproduce, to replicate. Okay, it's like we're going to go in there, use the cell's resources to make lots of copies, and then we'll bust out of this cell, probably killing the cell in the process, and then disperse in the bloodstream and attack other cells. So that's usually a strategy that viruses like to employ. Now, the antibodies can bind to the viruses and prevent them from entering cells. So prevent them from infecting the cells and then replicating themselves as well. So that would be the neutralizing properties of antibodies. Okay, when they do that, we call them neutralizing antibodies. So that's essentially how these antibodies work. Okay, now these B cell receptors, though, they have to recognize this. So right now they're tethered, they're not released in the bloodstream yet. They're tethered to the surface of the B cells, and let's say it meets its cognate antigen and then recognizes some binding sites, the epitopes, right? And then it binds. It still has to be activated, okay, before we can start to have it go into action. So the B cell receptors have these proteins that are associated with them, accessory proteins, and in particular we have Ig alpha and Ig beta, and they're right next to the B cell receptor, or BCRs. They're right next to them, and they're in the membrane, and then they have a part that's sticking into the cytosol, which means now they can signal, right? The thing with the B cell receptors, the stem part of the B cell receptor, the stem part of the martini glass, right, is in the membrane. So it's transmembrane, and then it has a little bit that sticks into the cytoplasm, but it's a really short segment. So it's pretty much useless for signaling. So it's going to need some help. And it has these proteins, the Ig alpha and beta, to help them. So these proteins are associated with that BCR, B cell receptor. And they have bits that are sticking into the cytoplasm, which allows them to signal. Now, they can't recognize antigen, okay? It's the antigen binding part of the BCR that will recognize the antigen. But once that has been bound to an epitope, then we can use the Ig alpha and beta to activate and start the signaling process. The first thing we have to do is actually this process of cross-linking. It's a funny name because we're not really cross-linking anything, but that's just been the term applied to what happens. And essentially, it's clustering these B cell receptors together. So on the cell membrane, you have these B cell receptors, and there's sort of one here, one there, and they're dispersed throughout the membrane. But when we start to bind epitopes out there in the blood, then what happens is they start to cluster, and these B cell receptors are brought together in lipid rafts. Okay, and they're going to be in close association with each other. We call that clustering cross-linkage, even though there's no real cross-linkage going on, all right? And you have, different pathogens out there with the epitopes. Each epitope is being bound by a B cell receptor. Sometimes a pathogen will have repeating epitopes or multiple epitopes. So on its structure, maybe on its cell wall or something, it has a certain protein sequence that repeats. That's the sequence that actually binds to the B cell receptor, but it has a number of those sequences, right? So now we have clusters, and we can have different B cell receptors. We have many B cell receptors on that cell membrane, but now as they're recognizing the epitopes, they cluster and do this cross-linking process, okay? And that's very important because that tells the accessory proteins, the Ig alpha and beta, that, okay, we've engaged the enemy, okay? So now the B cells themselves have receptors that can also bind complement fragments. Remember from the previous episode, the complement system, right? We have these complement proteins and they're split up into fragments. And these fragments, some of them opsonize the pathogen, so they kind of tag the pathogen to tell the phagocytes to kill it, right? So these B cells also have receptors that can bind some of these complement fragments that are tagged to a pathogen. And so we can actually bind to that complement fragment, and that would be called a complement receptor on the B cell. It's different from the B cell receptor. This is not that antibody that's tethered. It actually recognizes the complement. So it's called a complement receptor, and we call this a co-receptor, okay? Because when you have the B cell receptor engaged, bound to epitopes, right? And then you also have these complement receptors on the B cells that are binding to the complement fragments that are attached to the pathogen. It kind of clusters all of the B cell receptors together. And this kind of amplifies the activation signal. So you actually don't need as many B cell receptors clustering to activate that cell. Okay, so it's a useful way to amplify the signal transduction. Now, a B cell that's never encountered its antigen, that's a naive B cell. But once it has encountered its antigen, then we call it an experienced or mature B cell. Now, this experienced B cell, it has been activated either by a T-dependent pathway, T-cell dependent pathway, or T-cell independent pathway. So there are two ways that we can actually activate the B cells. So when you cluster the B cells together, we need yet another signal, a second signal that's a co-stimulatory signal from T helper cells. Okay, they are also part of the adaptive immune system. We're going to talk about them in a separate episode. But we have these T helper cells that will co-stimulate these B cells. All right. Now, most commonly, we know that B cells have a protein called CD40, 40, on their surface. Okay, and these T helper cells have CD40L or CD40 ligand that can bind to CD40. Right? So these T helper cells, when they see CD40 expressed on the B cells, they will bind their CD40L to CD40, and this is how they will activate those B cells. Okay, so that second signal has to come from the T helper cells. That's the T cell dependent pathway. The independent pathway, you just have repeated epitopes. Okay, and a lot of them that the B cell receptors are binding, and they're binding a lot of these episodes, and there's this mass effect of bringing the B cell receptors together in a cluster so that they have this cross-linking procedure. And then the second signal is actually a danger signal. Okay, if you remember the toll-like receptors that we talked about in the previous episode, the TLRs. Okay, so these B cells also have TLRs, and that's the second signal that could be activated, right? So now they have a lot of epitopes, repeated ones that they're binding to. There's a massive clustering, and then the second signal, instead of the T helper cell co-stimulation, is going to be this non-specific danger signal. So one of the toll-like receptors, for example, gets activated, and now we can activate that B cell in a T cell independent manner. And this happens a lot with B cells that are found in the spleen. And the advantage of that is you get a quicker response. You don't have to wait for the T cell co-stimulation, right? So there's a bit of an advantage to that because those B cells can start responding quicker. And we see those types of B cells particularly in the spleen, and they act against organisms like Streptococcus pneumoniae, which is an encapsulated organism. And that's why if someone has had their spleen removed, then they're more susceptible to infections by encapsulated pathogens. Okay, so that would be the two ways to activate the B cells. There's another way, and it's kind of a bit of a trick. It's called polyclonal activation, and it really doesn't depend on antigens at all. This usually we see with parasites. Okay, so parasites, they can have some molecules on them that bind to other molecules on the B cell that are not part of B cell receptors. Okay, so these pathogens just have substances on them that are able to bind to some other molecules on the B cells. Okay, so these molecules on the parasites, we call them mitogens, mitogens, mitogens. Okay, so mitogens can do that, and they'll bind to these other molecules on B cells, and then it brings all these molecules together. We start to clump those molecules, and then we kind of drag the B cell receptors along. Okay, so it's not that the B cell receptors are binding to the mitogen. They're not. Okay, but they're part of this mass effect because as these mitogens bind to these other molecules on the B cell, and these B cell molecules start to cluster, they kind of drag along the B cell receptors. And so as a result, we get clustering of the B cell receptors as well, and they end up in a clump. Okay, now the thing about this is you are able to get different B cells to be activated by a mitogen because after all, we're not recognizing an epitope, right? It's not the epitope that's being recognized, it's other parts of the parasite, these mitogens that are kind of attracting other molecules in the B cell. So you can have different B cells with different B cell receptors that actually don't recognize an an epitope, and they get clustered and they get activated. And what's the point of that? Well, it's no good for us, right? But it's really good for the parasite. It's actually a strategy that they're using as a decoy and distract method. Okay, so what they're trying to do is, we'll get all these different B cells activated and engaged, and they're not specifically going to recognize us because we're not doing that through their epitopes, through the B cell receptors. So we kind of waste their energy and resources that way. Okay, so it doesn't work well for us, this polyclonal activation, but it's a good strategy if you're a parasite and you want to kind of, hack the immune system in some way. All right, so now let's talk a little bit more specifically about the classes of antibodies. Now, a naive B cell that hasn't seen its own antigen yet, it's going to mostly produce IgM, okay, and some IgD, but mostly IgM, okay. So this is the first antibody class of antibodies that is produced. So next to the gene sequence that's encoding for IgM are gene sequences for IgG, IgE, IgA, the different classes of antibodies. So to switch classes, to go from IgM to another class of antibodies, you just cut off the IgM part and you put in, let's say the IgE part or the IgA part, and you would make that corresponding antibody. Okay, so the antigen binding site, the FAB, is still the same. We haven't changed that. We changed the FC region, the constant region, okay? The region that binds to FC receptors. We changed that region, and it's important to note that that part, the FC region, is what determines the function of the antibody. That's the important part of that. So let's take a look at IgM. For example, that's the first, antibody that's produced, okay? And IgM is, I always think of M for massive because it's big. It's big, and it's sort of a pentameric structure because it's essentially 5 of those martini glasses combined together. And all the stems are pointing in, and then the mouth parts are pointing outwards. And if you can visualize 5 martini glasses arranged that way, that's essentially, how you could think of the IgM structure. Okay, so it's pretty big, and it's really good for activating complement. If you remember when we talked about the complement pathway, we talked about the classical pathway, right? And I said that's activated by antibodies. Well, here we are, right? So this IgM is great for activating complement. Remember the complement proteins, okay? They associate in a complex called C1, and that's a pretty big complex. The problem is it can't actually initiate its cascade down that complement system because we have these inhibitor molecules that are bound to it, right? So these inhibitors are bound to this massive C1 complex, and we really cannot initiate the complement cascade. But when you can clump together 2 or more of these C1s, if you can clump them together, then the inhibitor molecules kind of drop off. Okay, they just aren't able to bind, and they drop off. And once they do that, you can activate that complement system. Now, if you think about it, what I told you is that the IgM can bind to the complement. So what it does is that you have this binding to C1, right? And as a result, we bring, because it's got 5 of them, remember, you can bring, for each FC region can bind to that C1 complex. And you have 5 of them. So you can bring multiple C1s together, and when they clump like that, you're going to get rid of the inhibitors, and then we can activate that whole complement system. Okay, so that would be the classical pathway. So we can see that that's useful because in the beginning, as the complement system is trying to help rev up your immune system, that's part of the innate immune system. We can see that IgM can also get into the picture by sort of, activating the complement pathway. Okay, so another route for activating complement. We can look at IgG now, and these, by the way, are written capital I, small g, and then the M would be capital. Here, the G would be capital, okay? So immunoglobulin G or immunoglobulin M really is what they are. So IgG, it can also fix complement, but, it's only got one. It's sort of on its own. Remember, IgM has this cluster of the 5 martini glasses. IgG is just one martini glass. It's got one FC region, and that can fix complement, but you can't get the complement complexes to clump and then drop off the inhibitor molecules, right? So it's not that useful. You can only do that if, let's say, you have, a lot of the IgGs around, and then a lot of the complement around. So just, in close approximation, they can happen to do that. So they're not as effective, let's say, as the IgM would be for activating complement. But, IgG has other functions. So for example, there's a subclass of IgGs called IgG3. And here, the importance of this is it can work with natural killer cells, the NK cells of the innate immune system. All right. So now you're starting to see how, yes, we talk about the innate system and then we say there's a separate system, the adaptive system, but you really see how they work together. Okay. Because these NK cells have receptors for the constant region on IgG3. Right? So they have receptors for FC on IgG3, and therefore they bind to IgG3. Now, the FAB or the antigen binding site of IgG3 is still open. When that binds to the pathogen, right, then basically the natural killer cell is directed to killing that pathogen. So the natural killer cell doesn't recognize antigen, but it can bind to IgG, and IgG can recognize antigen, right? So they kind of work together and they have this collaboration. So I'll pick out who you should kill, says IgG3. Oh, that one. Okay, I've bound to it. Now you bind to me and we'll put you guys together and gobble that pathogen up. Okay, so that's what it can do with the natural killer cells. And also IgG can cross the placenta, so it's useful because it can help protect the fetus, right, from infections. It's got a longer half-life than IgM. IgM is going to hang around for a day, or the half-life is about a day, and IgG3 is a couple, 2 to 3 weeks, 3 weeks maybe, for IgG3. Okay, so that's handy. Now let's look at IgA. IgA is interesting. It's secreted by the B cells at mucosal surfaces. So oftentimes people think of IgA in the gut, obviously, because, that's a mucosal surface. But we also have, let's say, the mucosal surfaces in your respiratory tract. So that's another place you're going to see IgA. Okay, so these are immunoglobulins secreted by those B cells at these mucosal surfaces. And then they have a dimeric form, so the stems of the glasses are kind of, held together. And this allows them to go, let's say in the intestines, it allows them to pass out of the intestinal wall into the intestinal lumen, okay? The part of the intestines where the food and the microorganisms hang out, okay? So we can get that IgA out into the intestinal lumen, and it's useful because it's resistant to digestive enzymes or to acids. So it's kind of resilient, and it's able to clump pathogens and then have those pathogens, be passed out together with poop. Okay? So that is a good way that IgA can protect, the body at mucosal surfaces. And it's also found in breast milk. I just wanted to add that. Then we have IgE. Okay, IgE is useful against parasites. Okay, I think we always used to say worms, wheezes, and weird diseases. Okay, I think that was the mnemonic. Anyway, we have white cells. I didn't talk about them much, I don't think, called mast cells, right? And mast cells have these granules in them that have a lot of toxic chemicals in them. And when they encounter a pathogen, especially parasites, then what they do is they degranulate and they dump these toxic chemicals onto those parasites in order to irritate them and maybe to destroy them, okay, or take chunks out of them. All right, part of those chemicals, we have histamine as well, okay? Histamine is one of the very well-known components of mast cell granules. Okay, so we have mast cells, they see parasites, and they dump their granular contents on the parasite, and that's the first exposure. Your body also makes IgE to, bind to the parasites as well. Okay, that's all well and good, except that you can also have your mast cells binding to the Fc portion of IgEs. Okay, so you see all this IgE floating around the mast cell. It has receptors to bind the Fc portion of the IgEs. Okay, now we have a mast cell there that is studded with IgEs on its surface. Okay, this is going to lead to problems because the next time you're exposed to that pathogen, to that parasite, let's say, then you're going to have a massive reaction because you have all these IgEs now on the surface of the mast cell. We're going to have a massive mast cell reaction, and all these mast cells are going to just explode and release their granular contents, right? A large amounts of histamine. And this is what we see in an allergic reaction. So in this case now, the antigen becomes an allergen, right? And we cross-link a lot of these IgEs that were on the surface of the mast cell. We cluster them together. You have this signal for a huge mast cell degranulation signal, and we dump all this histamine out, and this will, for example, cause the capillaries and blood vessels to be leaky, so there's a lot of fluid accumulation and so forth. We can also cause smooth muscle to contract. So if you have the smooth muscles in your respiratory tract and your airways and they constrict, that's going to cause respiratory difficulty. All right. So IgEs, yes, useful in parasites, but also prominent when we see allergic reactions. Okay, class switching. We talked about how we can switch from IgM to IgG or IgE and so forth. It tends to be controlled by cytokines, and these are chemical signals that are produced by those T helper cells as well. So again, when we talk about the T cells, we'll go into this a bit more, but you'll see that the B cells, they don't function alone, and they need the help of the T helper cells. So as an example, the T helper cells maybe are producing a lot of IL-4 and IL-5 in the presence of parasites. And so now we switch. Those cytokines will prompt switching of IgM class of antibodies to IgE. Okay, and that would be relevant in that specific situation and context. Interferon gamma will cause switching from IgM to IgG3. TGF beta, that will cause switching from IgM to IgA. So you can see these cytokines that are produced by the T helper cells are going to be important in class switching there. Okay, last thing just to say is that these B cells, there are essentially two paths for them. One is that once they have recognized the epitope, they've been activated, they can become plasma B cells or they can be memory B cells. So the plasma B cells are essentially just going to crank out antibodies. So they'll make these antibodies, crank them out and release them into the bloodstream to help fight the pathogen. And because, it's very energy intensive and it's sort of this huge production unit, then those cells, actually, the plasma B cells don't live very long. But while they're alive, they're really pouring out the antibodies to help in our body's defense. They can also, some of them, they will become memory B cells. And these memory B cells, basically they're long-lived and they hang around because the next time you get infected, let's say by the same pathogen, they already recognize the epitopes on those antigens and they can very quickly now crank out those antibodies, right? That's essentially the whole idea behind vaccines and making antibodies, because now you have these memory B cells that are able to crank out the antibodies in a very short period of time. Okay, so see, we only talked about the B cells. If we tried to go into the T cells, this would be like a sort of a ring cycle, a Wagner ring cycle or something like that. Okay, so anyway, we can go into our wrap up now. We talked a little about the structure of the antibody, that whole martini glass thing, and I talked about the different regions. We talked about the antigen binding region, and then, which is, essentially sort of the mouth part of the martini glass, and then the stem part is the constant region, which determines the function of the antibody. How do we get such diversity? Well, we have this mix and match strategy of different gene segments, right? So we have the VDJC segments that can be rearranged and mixed and matched to form, different light and heavy chains. And once we have a particular light heavy chain that's viable on the cell surface, right, then we have a specific B cell receptor on that cell surface. And we have one B cell receptor per, one type of B cell receptor per B cell. But that B cell can crank up many copies of that B cell receptor and then stud the surface of the cell with those copies, okay? But it's that one type per B cell. At first, you don't make a lot of copies. You just kind of make a few. But once you have a B cell receptor recognize an antigen, then it starts to proliferate. So we now make a lot more of these B cells that can then crank up, make those B cell receptors and antibodies. Now, the antibodies, we said, don't actually kill the viruses or bacteria. They opsonize them, they mark them and tag them for destruction, or they can neutralize them by preventing them from entering cells, let's say viruses from entering cells and replicating within cells. Okay, we have to have signaling, and so for that, we need the help of these accessory proteins, Ig alpha and Ig beta, that are associated with the B cell receptor. Okay, so the signaling can happen with the B cell receptors recognizing epitope and lots of epitopes, and then clustering in these lipid rafts, okay, forming these clusters, and that would be called cross-linkage of B-cell receptors. But we also have complement receptors that recognize the complements that are tagging pathogens, and these are co-receptors, right? And so together with the B-cell receptors, we can amplify that signaling pathway, right? You could just recognize via B-cell receptors alone, but you have B-cell receptors and those co-receptors via complement that's tagged already to bacteria or viruses, right? Then you actually amplify the signal, and you need less of the clustering to happen to activate. Now, activation of the B cells can occur in a T-dependent or T-independent fashion. With the T-dependent fashion, the B cells, for example, will have proteins on them such as CD40, and that will bind to CD40 ligand on T cells. And once we do that, we can activate the B cells. We also have T cell independent pathways, and this is just lots of repeated epitopes on the pathogen, right? So that we cluster, we just activate, we just bring together a lot of these B cells that are binding to those epitopes. And that way, plus the addition of a signal, a danger signal, such as activation of toll-like receptors, that's going to be able to activate the B cells independent of T cells, without T cells, okay? And then we also talked about this trick, this polyclonal activation that parasites like to use, where they have mitogens that are recognized by molecules on B cells that are not the B cell receptor, right? And because they're not specific, they're not B cell receptors, they kind of draw a lot of different B cells to them, and a lot of different B cells will bind, and this acts as a way to distract the immune system, okay, and spend its or waste its resources. Okay, then we talked about class switching and how we always start off generally with IgM, which is, sort of a huge, massive complex. You have a pentameric structure where you have 5 of those martini glasses arranged such that the stems point inwards and the antigen binding regions point outwards. Okay, so that would be the IgM. Now, if we want a class switch, we just, in the gene segments, cut out the IgM part and then put in whichever class you want, IgG, IgA. Okay, so that's how we class switch. The antigen binding portion is not changed, just the constant region is changed, which is important because that is what determines the function. So we talked about how this massive IgM complex can actually bring these complement sort of complexes together. C1 complement complexes together, they're very large, but if you bring them together, then their inhibitor molecules are dissociated from the complex, and we can activate the complement pathway that way, and that would be the classical pathway to activate the complement system. IgGs, they can fix complement too, but because they only have, per IgG, they only have one constant region, so they can't really easily clump those complexes, the C1 complexes together, unless there's a lot of IgGs and those complexes around, and just by being in close proximity, right, we do that. But it does potentially fix complement as well. But IgG can bind to the natural killer cells, right? So natural killer cells, they actually bind via the Fc receptor to IgG, and the IgG antigen binding site will recognize epitopes, bind to the antigen, which is the pathogen, and then kind of direct the natural killer cells and say, this is the cell you're going to kill. This is the cell you're going to gobble up. So that's a nice little bit of sort of cooperative work between the innate and the adaptive immune system. And in addition, that IgG is useful because it can cross the placenta. IgA, dimeric, you have sort of those two martini glasses with the stems, touching each other, and then the glass portion, the mouth portion facing outwards, right? And they're secreted by the B cells in mucosal areas of the body, like your gut and like your respiratory tract, and that it will be able to be secreted into the lumen of those tracts, and they can clump pathogens and cause them to be removed in mucus and in poop, if we're talking about your intestinal tract. Now, then we also mentioned briefly IgE and how that's used a lot of times when we encounter parasites in our body, but also they're associated with mast cells. Mast cells degranulate to attack parasites, but they can bind to the Fc portion of IgE. And if they bind to a lot of the IgE and they clump the IgE together when they're next exposed to the antigen, we can have a massive mast cell reaction, degranulate, drop a lot of histamine and other toxic chemicals, and cause an allergic reaction, an anaphylactic reaction even, right? Depending on the scenario. And then lastly, we just mentioned how the T helper cells are really important in class switching because they secrete certain cytokines that direct the class switching. Okay, made it through the B cells. Like I said, the immune system is massive. And, excuse me, I naively thought I could tackle it all in 1 or 2 goes, but then I realized, nope, I need something to drink. I think I'm just relieved that I got through the episode. I apologize. I hate coughing into the mic, but there's no way I can cover it. Anyway, I hope that was a helpful episode for you. Please check out my website, vivianlohmd.com, VYVYANELOHMD.com. Send me your requests, your comments. Check out the live stream event dates. Subscribe to the newsletter. I do put upcoming episode topics on the newsletter and sometimes talk about projects that I'm working on. And yeah, I hope to see all of you at a future live stream event. But for now, we made it through another episode. So signing out from VLMD Rounds, I'm Dr. Vivian Loh, and I sing the body electric.